Thank you for the kind introduction. I would also like to thank you and Sanja and all the organizing team for giving me the opportunity to be part of this workshop. It's just a stimulating program. I was privileged to co-chair the 50th workshop with Claude Bachmann, which seems like it happened just yesterday. And that time we thought, wow, 50 workshops already. What a success. And now it's the double number almost, 99. It basically shows that these workshops have something very special to offer, with always great programs, with a mix of speakers from very different backgrounds, and as Hanya already said, with lots of room for interaction and discussion, which makes it very special. So I'm really grateful for the opportunity to participate here. However, the price I have to pay is that I got a topic that is impossible to cover in half an hour. It is mechanisms of nutrition programming, which is a topic where we had huge amounts of research over recent years, and I cannot possibly cover the developments that have taken place, which are very exciting. So we just try to, do you still hear me? Yeah. Try to scratch the surface and give you a few headlines on where we are. I don't declare a conflict of interest, but I'm grateful, of course, to the National Nutrition Institute for supporting this event. I'm sure you all, and every one of you have heard about the concept of early metabolic programming of later health. So we have seen so much evidence now that environmental cues, including nutrition, during sensitive but limited time periods of early development, induce lasting effects on our body, the structure, the physiology, health, performance, and disease risks lifelong. We often talk about the first 1,000 days of life, which we consider a particular sensitive period of pregnancy in the first 2 years, but we know it's not exactly 1,000 days. For example, preconception is also an important window of opportunity for metabolic programming. And we are particularly interested in the application to improve population health. We all know we are in a state of the world where non-communicable disease burden explodes. 3 of 4 deaths around the world are due to NCDs, which a large portion contributed by cardiovascular disease. We know prevention is possible in adult life, but we have enormous opportunity to contribute to risk reduction in early life, and this is what motivates many of us to invest further in this area. Now, I strongly believe if we want to be successful, we need to understand mechanisms so we can develop better preventive approaches. And I would like to focus on the first 4 here on this slide: growth and structure, epigenome, metabolome, hormones. I will not touch upon microbiome because there will be separate presentations on that topic. So let's start with growth and body structure. This is what we believe today are key pathways that link early growth trajectories to lifelong health, particularly in interaction with the current environment, lifestyle, in adulthood, and our genetic makeup, to increasing the risk for obesity, adiposity, and related NCDs. So we are concerned about rapid fetal weight gain, we are concerned about rapid postnatal weight gain, and particularly concerned about the mismatch of slow fetal growth with low birth weight followed by rapid postnatal growth, which is happening in many underprivileged populations around the world. So let's look at some of the data. We have known for long that children who have a rapid weight gain in the first year of life, in the second year of life, have an increased risk of obesity later. You see here on the left a number of observational studies that show in totality the adjusted odds, adjusted for confounders, of obesity in later life, in childhood, adolescence, adulthood, is almost doubled if you have a high weight gain in the first 2 years of life. And the study on the right shows basically the same. Compared to those who don't have a rapid weight gain, those who have a high weight gain, here expressed as high BMI gain, early in life, in the first years of life, have a lasting elevation of their BMI trajectory in adolescence, they are on a road to disaster with more overweight, obesity, and the adverse consequences thereof. Now I got stuck here. Now here we are. So you may say, well, this is only a small number of children who have a rapid weight gain. That's not the case. We have looked at this in this analysis of growth trajectory in 6,700 children from 11 countries, all affluent populations. And you see the green growth curve, a body mass index standard deviation score of 0, is only half of our children. So what we think is normal growth along the percentile is only every second child. Almost every second child in the blue curve, 48.5% of our children, have what we just seen, rapid weight gain the first 2 years of life, where the BMI curve goes up, then it goes down a bit more towards early childhood, but it remains elevated. So every second child is in this risk group that has an increased odds of obesity. Unfortunately, only a very small number, 1.5%, is in this extremely high-risk group who go up with BMI and go up like a rocket. I will show you later it's related to early nutrition. Now, this has lasting consequences. In this study, we looked at outcome at 20 years in adulthood, and you see that the early growth trajectories are related to an increased risk of high BMI, high body fat mass, high fat mass index. In other words, the predictors of diabetes, cardiovascular disease, some forms of cancer. Why is that? Well, if children have a high weight gain early on, they don't deposit lean body mass. You cannot build so much muscle in a short period of time. If you have a rapid weight gain, you will deposit more body fat. You will produce children that have a greater proportion of body fat, which then reduces insulin sensitivity, increases the risk of insulin resistance, along with the adverse metabolic consequences and cardiometabolic consequences that we all know are related to insulin resistance. So what we can conclude today is high early weight gain promotes the risk of increased body fatness and obesity. So we should aim at preventing high early weight gain. We should support families to move the growth trajectories closer to the WHO growth charts. But we have a lot of open questions. We don't know what growth trajectories, what patterns of body composition promote optimal health, under which conditions. We would assume, what we don't know for certain, that different groups of children and infants have different risk status. Is it genetic? Is it other biological? Is it environmental determinants that we need to consider? We also don't know what are the most effective interventions, during which time window and in which subgroups of our childhood population. And of course, we want to know what are the effect sizes that we can achieve and how can we best achieve post-benefit. Now, nutrition matters for early growth. We all know that. And if you look at the study that I showed you before, children that are not breastfed or breastfed for short have a much increased risk to be in one of the two top growth trajectory groups where you have a higher adverse effect. So if you're not breastfed, you have twice the risk of early rapid growth, 2.5 times the risk of this red curve with extremely high likelihood of obesity. Early feeding seems to matter, and that was shown in many observational studies around the world, that you all know breastfeeding is associated with a moderate but consistently shown protective effect against later obesity. In these 5 meta-analyses, the effect sizes were between 12 and 26%. Not huge for the individual, but a huge benefit for population. If we can prevent every 4th or even every 8th or even every 4th case of obesity by improved infant feeding, imagine what the benefit is for population health and also for the cost of healthcare. That's a huge benefit. So what is on the poster here on the right is still true. Nurse the baby, it's your protection against trouble. And I really like the last line, consult your doctor. Don't just trust the internet. Ask the doctor about infant feeding. An interesting observation also is that dose matters. And everywhere where we have a dose-response relationship, we think causality is plausible. So this meta-analysis, like others, has shown the longer to breastfeed, the more the risk of obesity later is reduced. And it appears to be the total volume, the total amount of breastfeeding, not so much whether it's exclusive or non-exclusive, that has no significant effect, but really the dose of breastfeeding overall seems to be related to the outcome. Now, a caveat is that these are all observational studies, and clearly residual confounding is likely. Most of these studies are from high-income countries where higher socioeconomic status is associated both with more and longer breastfeeding and with better health. So maybe the true effect size of causal effects of breastfeeding is smaller than what we see in the studies, but the results are so consistent that I think it still gives us a lot of argument to do what Professor Shajewska said in the introduction, do everything we can as healthcare professionals to enhance promotion, protection, and support of breastfeeding. Questions for further research include, how does it actually work? Why is it that breastfeeding modulates growth in body composition, not only during the time of breastfeeding, but even years later into adulthood? How can that happen? What is the mechanism by which the body remembers how it was fed in the first year of life? And which infants and population groups are highly susceptible? And finally, we are not really good at knowing what interventions work. If you look at the literature, breastfeeding interventions to promote intervention, we have probably a lot of room for improvement, for developing further strategies and for evaluating them. Epigenetics is another mechanism that has received a lot of attention in recent years. Epigenetics is hereditary cell characteristics inherited from one cell to the next generation, but that are not determined by our genome, not determined by DNA sequence, but mechanisms such as the methylation of DNA, histone modification, or non-coding RNAs. The one concept that we have is that particularly during early life, environmental cues, including nutrition, change through epigenetic mechanisms permanently or for a lasting period at least, the expression of genes, and therefore with the same genotype, we can end up with very different phenotypes with very different patterns of disease risk depending on epigenetic modification of gene expression. The one mechanism that is most studied is DNA methylation because the technology has moved forward a lot. It basically measures how much methyl groups are linked to cytosine, one of the 4 letters of our genomic alphabet, always linked to guanine. And if we look at the distribution of these CpG sites, most are dispersed all over the genome and we don't know what they do, but a small percentage is located in the regulatory regions prior to exons. So if these CpG islands are not highly methylated, or have a low degree of methylation, transcription factors can bind and the gene is expressed. If there's a high degree of methylation, the binding of transcription factors is inhibited and genes are less or not expressed. So it's a very effective mechanism, particularly since the DNA methylation pattern is inherited from one cell generation to the next during mitosis. I can't give you all the data because time doesn't allow for it. I just give you one example here from our own studies where we see that the DNA methylation pattern predicts child fatness in preschool age in children of the European CHOP study. And what we are particularly interested in is, of course, the role of those genes that are switched off, if you like, by DNA methylation. What is their physiological function? How are they related to the pathways that lead to fatness? Because it's not a epigenetic DNA methylation all over the genome, specific sites that are responsible for the association. The role of epigenetics is now supported by many experimental studies, animal studies, and a number of human observational studies, but we have much less evidence from controlled intervention. So we have a number of questions still, how relevant that actually is in humans, how much causality we have here. We know that genotype variation may matter, so we need to understand this better, what the interaction of genotype and epigenetic marks is, which are the functions and pathways that are altered by imprinted genes, and how can we establish causality, causal effects of epigenetics on clinical endpoints, how we corroborate this in randomized trials. Metabolism is closely linked to nutrition, we all know that, and it is linked to observations on programming effects. We talked before on rapid weight gain as one of the predictors of long-term health, or disease rather. And for example, here in this group of 700 plus infants, again from the European CHOP study, when we look metabolomic characterization of the biochemical status of the infants, we see there's a number of metabolites above the Bonferroni line that are significantly associated with rapid weight gain in early life here in the first 6 months. There's amino acids, and there's a number of lipids, and the one metabolite that sticks out, that has the closest relationship, is lysophosphatidylcholine with myristic acid. It's basically a catabolized product from lecithin. Lecithin has 2 fatty acids, one fatty acid split off, often an omega-6 fatty acid, and you end up with this phospholipid with just 1 fatty acid, which is myristic acid. And we see this again and again popping up in different studies. Also, when we look at long-term outcome, it's the one and only biomarker that we find to predict obesity at school. Wonderful if we could reliably detect those children that have a high risk of obesity later at an early point of life, and then can do very much a targeted and focused prevention for this high risk. We are not there yet, but we would like to move into this direction. Now, we talked about the protective effect of breastfeeding. What we have learned is that not all breastfed infants are equal. We looked at here the metabolism of breastfed babies at the age of 6 months, 183 fully breastfed babies, and if you look at the metabolite patterns, there's 20 completely separate clusters of metabolite status. And what's more exciting, if you look at the right panel, for the 4 dominant clusters, the metabolite pattern at 6 months doesn't discriminate between growth at 6 months, but it predicts growth until school age. Isn't that amazing? If you have the metabolome pattern at 6 months, you can predict the likelihood of overweight at 6 years. So exciting to learn that although we think breastfeeding is always the same, it's not. There is a lot of variation between breastfeeding, and there we also know there's a lot of variation among the milk of different mothers. Infant sex also plays a role. This is just one example of many examples where we see that metabolites predict birth weight. Here, for example, omega-3, omega-6 fatty acids. But in red are the girls, in blue are the boys. The power of the metabolites on predicting birth weight is stronger in girls than in boys at birth. We don't understand this why. We see this also in childhood, we see this in adolescence, we see it before puberty, before a lot of hormonal effects, and we yet still have to understand what the physiological background is. But it basically tells us biology is always more complicated than we would like it to be. Now, I told you about lysophosphatidylcholine with myristic acid. If we look at it in the serum of babies, we see that it is predicted by the protein content of human milk. And by the way, also the insulin levels in the blood of babies is predicted by the amount of protein they get from human milk. So again, breastfed infants are not all equals. It's a heterogeneous group of human beings. Now, protein also was shown in this study by Philippa Prentice to be related to infant growth. In this group of 600 plus mother and infant bears, human milk composition was measured at one single time point, 1 to 2 months after birth. And you see the more milk protein there was in the breast milk of the mother, the higher was the body mass index 10 or 11 months later. Isn't that amazing? A single human milk protein content measurement predicts your growth until the age of 1 year. On the right, the children that were not breastfed had actually a higher protein intake and also a higher BMI at 1 year. So again, breast milk is not breast milk, breastfed infants are not breastfed infants, and we still need to learn more about the physiological background. Now, following these and other considerations, we have done a large intervention trial to test whether actually modification of protein intake in infancy has a benefit. We gave we had a reference group of children that were breastfed exclusively for a minimum of 3 months and then further breastfeeding, and we had about 1,100 infants that were randomized at the median age of 2 weeks to infant milk formulas with a conventionally high protein content or reduced protein content, but the same amount of energy. And what you see here is that the mean body mass index was different depending on the feeding in the first year of life, when there was different feeding given. The breastfed infants ended up at a lower BMI than those fed the conventional formula, and the ones with the low protein formula were intermediate. Then at 3 years, there was absolutely no difference anymore. So if you would have stopped the study there, you would have said it doesn't matter. But then the curves deviate again, and at 6 years, those children who were fed high protein the first year were heavier than those breastfed or low protein formula. And what's impressive is that not all infants react equal, but those in the top percentile, here 90th and 95th percentile, show a much bigger effect of the early feeding. You see suddenly there, compared to the mean BMI, the effect size is huge. So again, breastfed babies and babies fed the intervention formula with less protein have the same outcome in terms of BMI, but those fed the high protein are markedly heavier. And that's why there's a big difference in obesity rate across the 5 countries where we did the studies. Breastfed infants, 3.6%, conventional formula, 10.5%, lower protein formula in this double-blind randomized trial down to 5.2%. If we adjust for other confounders, we have a 2.6-fold reduction of obesity risk at school age, but just by improving one element of infant feeding. Isn't that amazing? Bigger effect size than anything else we have today evaluated for obesity prevention in children. A really important benefit. And it extends until adolescence. The effect of the first year of live feeding is still visible in early adolescence. Now, why is it? We thought perhaps with a high protein, we enhance the plasma and tissue concentrations of those amino acids that stimulate the secretion of the key growth factors in infancy, insulin like growth factor 1, thereby promote higher weight gain, promote higher body fat deposition, and the risk of later obesity and NCDs. And yes, that's what we found. We found that the branched chain amino acids, the indispensable amino acids, differ in the feeding groups. They are lowest in the breastfed group. They're much higher in the group with the high protein intake. They are lower than that in the group that received less protein, but not quite like the breastfed group. The dispensable amino acids, however, were not at all different. However, when you look at the error bars here, you also appreciate that there's very large inter-individual variation within the group of children here. We think this might be important. We believe that this could be one element by which more rapid weight gain and body fat deposition could be triggered, because amino acids, along with the growth factors, stimulate mammalian target of rapamycin, which is a key sensor in each and every of our body cells that detects the metabolic status and then switches the cell to protein and fat synthesis, to anabolism and growth. We also looked at the effects in a study where we could do more measurements. In infant, you can only take one blood sample. So we used formulas with higher and lower protein content in adult volunteers. And you can see that with the higher protein supply in adults, you have an elevation of branched-chain amino acids that lasts for more than 5 hours. It's not a short-term effect. We also have an effect on insulin that peaks and goes down again after 1 hour. We have a response of the glucose curve. We have a response of the urea and triglyceride response at more than 3 hours after feeding. But again, if you look at the curves, large inter-individual variation, very large variation between individuals, which we don't understand. We gave the same amount of energy to the different feeding groups, the same energy in the formulations that we gave, and also we did a lot of detailed dietary measures, and throughout the whole study period, there was no difference in energy intake between the feeding groups. But the big difference in body fatness. With the high protein, you don't build your muscles like our adolescents believe when they buy these protein shakes, but you build more body fat. You become fatter with higher protein. Why is that? It's the same energy intake. It seems like implausible with thermogenetic laws. Well, we see indications that beta oxidation is inhibited by high protein intake if you look at the fatty acid acylcarnitine concentrations. So more of the fat is going into adipose tissue if less is going into oxidation. Animal and human studies identify metabolic predictors of health. Early nutrition can modify. What are the key pathways? How can they be modulated? What drives this large inter-individual variation? Can we identify and evaluate informative biomarkers? And can we design and evaluate precision nutrition interventions, their benefits and cost effectiveness? And finally, I want to have a short look at endocrine mechanisms. If we look at the same study, we see that in fact high protein elevates insulin secretion and IGF-1 concentrations, which we can reduce with less protein, although it's not becoming normal compared to breastfed babies, and the IGF concentration in the blood predicts early weight gain. It is a driver of weight gain. And if you look at what drives IGF, you can see genetics, infant sex have a very modest effect, but nutrition has a huge effect. We have a lot of potential for modulating the concentration of the growth factors. Interestingly enough, we also see again a very marked sex difference. Girls show a much bigger response to the dietary intervention comparing high and low protein with respect to total and free IGF, the IGF binding proteins, and also leptin. And again, we don't know why infant girls are so different from infant boys, what's behind this. If we look at trying to understand the relationship in the sparse model, we see that protein intake is driving the branched-chain amino acids, which drives the free IGF-1, which drives the BMI, standard deviation score. So it looks like we have really a chain of events that leads to increased obesity. And we're also concerned about the long-term impact. Many of you know that IGF-1 is stimulating malignant cell growth. Look at this study from the US, large study. If you're before 65 and you have a high protein intake, you have a higher risk to die, particularly from cancer, and you have increased IGF-1. If you get older, above 65, the effect of protein on IGF is attenuated and there's no more adverse effects or mortality, rather a protective effect. So young and old people, again, differ. Infant feeding affects endocrine growth modulators, large inter-individual variation. What are the key predictors? What is the role of not only protein quantity, but also protein quality? What are the most sensitive time windows? What are the drivers, predictors of inter-individual variation? And how can we design and evaluate better and targeted interventions? Finally, I would like to conclude with saying it is important that we invest in understanding mechanisms. It should lead to better strategies of effective health promotion. It should lead to more targeted interventions in susceptible subgroups that we call precision nutrition, and a greater efficacy, efficiency, and cost benefit of our preventive interventions. We are lucky enough that we can, with publicly funded grants, look at this a little bit further. We look at epigenetic mechanisms. We look at biomarker signatures, where we do what Hanya has suggested, use artificial intelligence, machine learning to try to integrate the data and try to improve obesity prediction and prevention. Thank you very much indeed for your kind attention, and allow me to say a great thanks to a wonderful team of colleagues and friends that work together on this topic at LMU in Munich. Thank you.